Thank you, everybody, for joining us this evening. This is another AONM webinar, and we're delighted to have Professor Leona Gilbert with us this evening, as well as Dr. Armin Schwarzbach. This evening's webinar is about quality. It's about the ISO guidelines and how they are revolutionizing Lyme disease testing. And I'll just briefly introduce the speakers. Professor Leona Gilbert is an adjunct professor of cell and molecular biology and the CEO of Tested OI. She has over 20 years' experience in research-driven initiatives that provide valuable tools for patients. She has a doctorate in biotechnology and very long experience in bioinnovation and entrepreneurship. She has supervised numerous PhD, MSc, and BSc students and published many peer-reviewed articles. So she'll tell us more about herself as she presents. I'll just go on to Dr. Armin Schwarzbach, who I'm sure you all know and most likely saw last week as well when he gave a wonderful presentation, a training for us. He's a world expert in the field of laboratory medicine and infectious disease. He's committed to working tirelessly on refining the various diagnostic tests for Borrelia burgdorferi, other co-infections and viruses. This is a research area to which he has devoted more than 20 years of his professional life. So he again is also open for any further questions about his background and experience. So let's dive right in. Thank you so much, both of you. They will talk for about 40 minutes perhaps in total, maybe a little less. Please put any questions that occur to you or comments in the Q&A or the chat this evening. And towards the end, we'll have lots of time for questions. So thank you very much.
Great. Okay, I'm going to share my screen here. And I trust that you guys can see it. And thank you, Jillian and Claire. And thank you, everybody, for showing up at this time in the evening. I do really appreciate your time. And I think that this is a really important and passionate topic area for both Armin and I. So full disclosure, as Jillian said, I am the CEO and co-founder of Tested. We are the inventors with my business partner, Kunal Garg, of TicPlex and Toxiplex. Okay, with that disclosure, it also highlights the fact that actually we are a medical device manufacturing company, medical device manufacturing. And this means that we manufacture in vitro diagnostic test kits, the test kits of TicPlex and Toxiplex. And I'm so passionate about this topic because there is so much inaccuracy when it comes to the narrative or how we speak or when we in the context of diagnostic kits. I'm constantly hearing that about the inaccuracy of Lyme disease testing. And frankly, this is actually not true. Yes, people have published and said that, the sensitivity hasn't changed in the last 20 years. It's from 60 to 75. But actually, if you look deeper into what's being offered and utilized for diagnostics, that's simply not true. Other narratives are that I hear, like there's a lot of false negatives coming from that test, or there's a lot of false positive. Oh, a positive IgM is just a false positive. And also I'm constantly hearing, even on websites of health authorities, that this statement here, that the Lyme disease test misses 60% of the cases, and that the tests are not validated. Constantly hearing these wrong narratives. And I implore you to think about it this way. We have the narratives of the inaccuracy of Lyme disease because the test kits are not being used appropriately. You're not going to test somebody that just received a tick bite for Lyme disease or borreliosis 3 days after they have been bitten. You're not going to use that test correctly. We're not going to use the test to actually test them, long-term for IgM in a very, very long-term situation, because most cases are that the IgM has gone down. So I want to employ to you to start thinking the alternative, that actually Lyme disease tests are accurate. And I implore you to look deeper into the instructions for use of these commercially available test kits, because the instruction for use is that legal document that have to demonstrate the sensitivity, the specificity, and the robustness of the test kit. So within this presentation, the goal is to demonstrate how ISO standards, so these are the standards coming from the International Organization of Standardization, how these ISO standards, these standards agreed by experts, okay, influence actually Lyme disease, and actually how the NICE guidelines from UK or other guidelines, how they actually interact together to demand quality that is offered commercially in Lyme disease testing. And when we go through these learning objectives, it's important to really think about the public health implications and the human rights aspects, because the patient deserves to have timely diagnosis with good quality test kits. They deserve and they have a right to get a timely treatment as well. And that starts from a diagnosis. And these in vitro diagnostic test kits are there to aid in the diagnosis of Lyme disease. So this is the goal of today's lecture. And again, I want to point out that these test kits are not notoriously inaccurate. I implore you that we're using them at the wrong stage of the diagnosis pathway. And if an IgM is present, don't by default say that it's a false positive. There's something going on with the immune system in that patient. And we do have documented cases and published cases of persistent IgM or IgG. But let's look at the NICE guidelines, and you guys probably are quite familiar with this. This also actually correlates well with the two-tier system offered in the United States. And basically the two-tier system is what most countries in the globe actually guide clinicians to use when deciding on which tests to offer in helping and guiding for diagnosis. So as we know that if a patient comes in and complains about that they have symptoms that could correlate to Lyme disease, and if they have an EM rash, that's a clinical diagnosis. They can get antibiotics and they are diagnosed with Lyme disease. But if they don't have an EM rash, in a lot of the cases, they don't. And there have been documents that say 50% of people don't present with an EM rash. Then the clinician may order an ELISA, the first tier. And the first tier will test, depending on the length of the symptoms, will test you traditionally for IgM and IgG antibodies in an ELISA-based test kit that are manufactured with purified or recombinant antigens. That means that they have actually tried to reduce the unspecific binding or the cross-reactivity that occurs in traditional ELISAs by offering and utilizing purified, okay, clean, and small peptides or small antigens, okay, in the test kits, okay? But these are derived from the VLSC protein of the Borrelia, or most normally actually recognized as the C6 peptide. Now, if that ELISA is not positive, okay, for either IgM or IgG, then they, in the NICE guidelines, will encourage you to retest after 4 weeks, because the individual needs some time to mount an antibody response against Borrelia if they are infected. So they encourage a repeat, okay, between 4 to 6 weeks. And that's really nice, because in some cases, in some guidelines of the Health Canada, we don't have this retesting or consideration of testing the patients after 4 to 6 weeks. But if the ELISA in the first tier, okay, is positive, then the NICE guidelines now suggest that you offer this immunoblot test, okay? And this immunoblot test is supposed to be more specific, okay, but there is no demand on which antigens to use, okay? And in the NICE guidelines, there is no demands of how many bands, 3 for IgM or 5 for IgG, traditionally seen in the American two-tier system. There is no demands yet on that. But if it is positive, then of course these guidelines suggest that you offer antibiotics. But if it is not, then the guidelines state that you should consider that clinicians should consider alternative diagnosis. But we know that there are exceptions to this. We know that seronegativity occurs in chronic patients or long-term patients. Okay, so what you're just seeing here in these NICE guidelines are for somebody that's newly bitten, that isn't chronically ill, that has a productive immune system, and they should actually be able to be recognized if they have an response against Borrelia by these two guidelines. But we do see persistent IgM and persistent IgG in individuals that have a compromised immune system. And we also, as I mentioned before, see individuals that are seronegative, so they're going to be negative on this guideline anyways, or the two-tier system. But because of this, there are other guidelines and demands that the NICE has, okay? And this is just a recap of what I just told you graphically, but the other guidelines that NICE demands is that these tests for Lyme disease have to be performed in laboratories that are accredited by UK accreditation services called UKAS. UKAS, okay? So UKAS has the power to accredit clinical laboratories against certain quality controls, and we'll talk about that in a second, okay? But also NICE guidelines, okay, guides and says that the clinical laboratory has to use validated tests. And the validation of these tests, okay, should include published evidence on the test methodology and its relation to Lyme disease and independent reports of the performance. Okay, and this is important to remember that the validation of the tests have to be documented, and you need to provide evidence, okay, and independent evidence, okay, that the test is working as it should and that it's validated, not only analytically, so how it performs, but also clinically, so how it performs in the clinic, okay? So with these additional demands, we're going to look at these a little bit later, and we're going to actually see how ISO standards actually fit in actually aiding the clinical laboratory in fulfilling these requirements of the NICE guidelines. And then there is one other demand that the NICE guidelines states. The tests have to be carried out in these clinical laboratories that participate in a formal external quality assurance program. And I do agree with all of these demands, okay, that NICE guidelines have actually stated here, because it ensures safety of the patient, ensures quality of the test kit, and ensures accurate diagnosis. So let's kind of like dive into this a little bit further, but I just want to point out this wording that the NICE guideline states as well. This is guiding clinicians do not routinely diagnose Lyme disease based only on tests done outside the NHS unless the laboratory used accredited participates in formal external assurance programs and uses validated tests. So this here is telling me that if a lab is accredited and a lab undergoes formal external quality assurance programs and uses validated tests, then it is okay. The clinician can routinely diagnose Lyme disease. And there are standards and agreements globally that UKAS actually signed to, okay, that actually allows this. And I want to bring that forward today. So that when we bring these documentation forward for you today, I will be able to give you kind of like the arsenal to go and say, hey, my lab results can be accepted, or my patient's lab from a foreign laboratory can be accepted here in UK or here in your own home country. It can be accepted. So we can start actually also changing the narrative that the Lyme disease testing is inaccurate, because it's absolutely not true. So one of the first ISO standards, okay, these international organizations of standards that experts agree upon is in relation to actually how the test kit is manufactured. So how the in vitro diagnostic test is manufactured. And this is the ISO 13485. It outlines the criteria and the demands of the quality management system, okay, for medical devices. And in this ISO standard demands analytical validation, demands clinical validation, okay, and also under the regulation here in Europe, this in vitro diagnostic regulation, it demands clinical utility. So fulfilling NICE's guidelines of validated tests, if this test is manufactured under this ISO, then that's fulfilled because the validation is demanded. It's documented, it's independent, and it's doing, of course, the analytical validation as well as clinical validation, and also then the clinical utility. And national medical device agencies like FDA, like I mentioned, the IVDR, in vitro diagnostic regulation in Europe, FIMIA here in Finland, DAX, they can also demand, okay, this analytical and clinical validation. And to fulfill those demands from those medical device agencies, of course, it's easy to demonstrate compliance against this ISO 13485 because it is demanded there anyways. So if we are audited yearly under this ISO, we show continuous reports, okay, independent reports as well of analytical validation and clinical validation, then we fulfill the NICE's requirement that the tests have to be validated. And this is what we're doing. These national medical devices will not accept sensitivities of less than 80% or specificities less than 80% for Lyme disease testing. FIMIA here in Finland will not allow this test kit to be sold to clinical labs if we have such poor analytical performance. Okay, so it's hard for me to believe that people are saying that ELISAs are only 60% accurate. Well, you're looking at probably old diagnostic kits that had poor performance, but look at the instructions for use, the legal document that this in vitro diagnostic demonstrates its performance, and you will see that we have great sensitivity and specificity in diagnostic kits nowadays because we're using really good innovative technologies like peptides, like purified antigens. So and better just, blocking solutions that prevent cross-reactivity with other species of Borrelia or other species of bacteria and viruses. So this ISO is very important for the test itself, okay? And you can actually demand this from the clinical laboratory. The tests that are using, are they manufactured under this ISO? And if they are, you should feel comfortable, okay, that it's fulfilling a global standard of quality. Okay, and it has been validated analytically and clinically. And so in relation to the clinical laboratory, this is the ISO standard that is very important to realize, okay? It is the 15189. Now this demands, okay, and the criteria for excellence and proficiency in that medical laboratory, and it guarantees the accuracy and reliability of its test findings. So it has all these demands to make sure that they are excellent in what they do. Excellent in performing those test kits, okay, that are manufactured under 13485. Excellent in actually processing, okay, your sample from the beginning to the end through its reporting of the results. The excellence here. So any clinical laboratory that you send out these test kits to, demand this, see and ask if they are adhering to this ISO, because this is the global standards, all right? And accrediting agencies like CAP, like UKAS, okay, provides certification, okay, to these laboratories, okay, to demonstrate conformance with this ISO. They actually use it, okay, hand in hand, ISO 15189 and the excellent performance that this lab is using. So they rely on it, okay? And under this standard, 15189, there is a demand of independently analyzing and clinically validating, okay, all medical devices that they bring into service. All the in vitro diagnostic kits, all of them have to have an independent analysis analytically and clinically under their own roof. They cannot bring any in vitro diagnostic kit under their roof unless they do this. So not only do we have the validation from the medical device manufacturer, but we also have the independent validation of these test kits under the laboratory that's using them. And this has to be documented. And you have to document everything in quality management systems because the auditors like CAP and UKAS can actually demand the proof of this, that you have done this and constantly doing this. Okay. So when they say that the test kits are not validated or the clinical lab isn't accredited, well, that's inaccurate too. It's wrong. You need to look and see. So we can start now by just demanding from the test kits and the laboratory, if they adhere and conform to those ISOs, we can start changing the narrative of these misconceptions and start speaking that actually we do have good diagnostic kits in Lyme disease. We're just not using them correctly. So if we go back to UKAS, okay, and the NICE guidelines, all right, let's put this, hand in hand. NICE says carry out tests for Lyme disease only in labs that are accredited to UKAS, okay, while the ISO 15 or 15189 is equivalent to what UKAS demands. So because of that, then that lab has equivalence to the standards that UKAS would audit for and to certify for. And UKAS has actually stated to me in an email that they can provide clinical laboratories that their accreditation by a national authority, as long as they follow the ISO 15159, is actually confirming with the UKAS standards. Okay, and I think Armin will talk about that in a second as well. NICE guidelines, remember on the second point here, they demand that you use validated test kits. Well, if the test kit is manufactured under the ISO 13485 and has the CE IVD marked here in Europe, so there is equivalent, okay? And this demand is fulfilled. In UK, the medical device and in vitro diagnostics and the regulatory medicines and healthcare products, the MHRA, they are the guidelines and the regulations that demand such performance of test kits, and they actually conform also and utilize the ISO 13485. So again, we have equivalence here. As long as that test kit, okay, is ISO manufactured under ISO 13485, has the CE and IVD mark. These are the legal marks that test kit can be sold here in Europe. And then the other demands of NICE that had was participate in a formal external quality assurance program. This demand is already there in the ISO 15189. So UKAS, when they go in, audit these clinical laboratories, okay, that is in a formal external audit, and that is their external quality assurance program that they use. CAP will do the same thing, and other accrediting agencies will also do external quality control audits. And audits are usually for us, for medical device manufacturing, it's yearly. And in some clinical labs, it's every 2 years, depending on who's the accrediting body. So I want to also put out there that there are these international agreements, okay, called the ILAC, so the International Laboratory Accreditation Cooperation Mutual Recognition Agreement, okay, it's a tongueful here, or a mouthful. So the ILAC Recognition Agreement is signed by 116 accrediting bodies from all over the world, okay, and the reason why they signed this, they wanted to have a kind of a national agreement that acceptance of this ISO 13, or sorry, the ISO 15189 can be accepted all over the world. So which means that if there is a clinical laboratory in UK, in Finland, in Germany, okay, in Australia, okay, accredited by these authorities, they can get their certificate that is issued by them demonstrates that they are fulfilling the ISO 15189 standard. And because of these accrediting bodies signed this mutual recognition agreement, that lab's accreditation should be accepted in any other country. Okay, and this agreement is only one of a few agreements that are globally signed with participating notified bodies or accrediting services that allows acceptance and equivalence, demonstration of equivalence, okay, and adherence to this 15189 standard. And this means that a foreign lab doing a test, okay, performing a test, if they are accredited by these notified bodies, like if there's a lab in Germany that's accredited by DAX, okay, and they also have, let's say, CAP or CLEAR accreditation, they've signed this mutual agreement, those test results performed under that lab should be accepted nationally and internationally. So this agreements usually facilitate global collaboration, okay, and guarantees that the labs are adhering to the most stringent, excellent criteria to deliver reliable and uniform diagnostic findings, and thereby actually enhancing the patient's care and clinical outcomes. So I brought up the case study of Armin's lab, and Armin will talk a little bit more about this, but I just want to put this on one paper for you on how I analyze these labs, okay, in regards to these ISO standards and these ILAC mutual agreements, okay? So Armin's lab carry out tests, okay? So the demand of these guidelines is carry out tests for Lyme disease only at labs that are accredited. Well, UKAS has provided, okay, a document that to show the equivalence, okay, of the standard that Armin Labs has because they are DAX accredited. DAX has signed this ILAC mutual agreement. And in addition, by the way, Armin's Lab is CAP accredited, okay, College of American Pathology accredited, and all of those actually demand the ISO 15189 equivalent standards, okay? And in addition, Armin's Lab offers tests of CE IVD mark products. That means that these tests are validated because they have been manufactured under a quality management system, and most of them under the ISO 13485. Okay, so the point is that for Armin's Lab, they are already participating in formal external quality programs, okay, which is demanded by ISO. It is also demanded by their CAP. So therefore, okay, any test performed under Armin's Lab, because of these accreditations and because of the DAX signing this mutual agreement, their test should be accepted if the test being used is CE IVD marked or manufactured under the ISO 13485. So this paper should be done on every single laboratory, on every single test kit, okay, to give evidence that you can walk in with and say, hey, you need to accept my test kit because of these points here. So why should any clinician or any health or health insurance refuse these test kits? Well, they shouldn't, because the international trust and recognition is actually under these two ISOs, the 13485 and the 15189. They are globally recognized for their reliability, excellence, and trust. Okay, they provide a consistent diagnostic quality, and especially with these mutual recognition arrangements such as ILAC. Okay, they have global equivalence. Okay, and because of this global equivalence, patients' outcomes can improve because there's timely and accurate diagnosis if foreign test kits are accepted. Okay, and also these agreements are in place and the standards, ISO standards are in place so that we can have acceptable international validated tests that can be utilized globally, and that will reduce the redundancy and minimize delays in diagnosis and aligns with global best practices and ultimately hopefully streamline healthcare delivery for global patients. So why accept foreign laboratories? Because acceptance strengthens countries' healthcare systems and reducing diagnostic gaps, promoting these international collaboration, and really fostering more innovations in diagnostic technology. And also, it's like there are human rights implications in refusing, okay, foreign laboratory tests, because denying recognition of these validated foreign tests, okay, can lead to delayed in diagnosis and treatment and probably infringe on the patient's right to health. So these tests, recognizing the tests, ensures equitable access, okay, of highly qualified diagnostics, regardless of the region, and aligns the country's commitment to uphold human rights and health equity nationally and internationally. So no laboratory or clinician should question the validity of Lyme disease diagnostics if the laboratory adheres to the 15189 and the IVD is manufactured, okay, under the ISO 13485 2000. Well, 2016 is the current standard right now, but they will change it. So I wanted to leave this with you here, and hopefully I have given you the kind of like the ammunition to walk in and say, hey, you know, accept these test results, okay? We're going to accept questions after Armin has provided his lecture, but I just want to end with this last slide in indicating that actually we are hosting a conference, the first annual conference of chronic infection pathology this September, and I encourage patients, clinicians and scientists all can come together to this conference and actually empower each other and learn from each other and actually on how we can help the patients that are really suffering from chronic infections. Okay, so thank you for your attention, and I will give the show to Armin.
Thank you very much, Leona. Yeah, you didn't mention that you were active in Australia, and you did a great job there with other experts fighting for the Lyme patients. Maybe you could tell us later on if also this quality management would be very important for the Australian health authorities. I think it is. Yeah, I want to start now with my first slide. Sorry, give me a second. In the end, I would say there's nothing to be added to Leona. Leona, it was fantastic. Thank you very much. I think to explain it in easy words, it's for you guys outside. You are not in laboratories working. You're not a test producer. Professor Gilbert, she knows both sides. She knows my side, and as a user, I'm a user of her test as an example, and her tests are CE certified and IVD registered according ISO 13485 from 2016. Maybe we mentioned that. We did the accreditation also in according 2016. Now we have a new accreditation standard in Germany. The 15189 was modified, and they are very closely now to the CAP accreditation. So we see the differences. And I would say three words are very important in this. It's to trust, or it's the trust of a laboratory test, or it's a trust of a treatment. It's a reliability, which is always a very sensitive word because they say this lab is not reliable, this test not reliable. So we could discuss later that. And the last point, I would say, it's safety. And why? Safety, because if you get a wrong laboratory test result, your doctor does a wrong therapy decision for you. And as an example, if a Western blot gives you a false positive IgM, the doctor says, oh, you have some Lyme similar symptoms, you're using antibiotics, you could be damaged. And this is exactly, we come to that point. FDA in America is on the same way we are in Europe. We could shout at them, we could say, oh, they are all Lyme denialists, but that's not the truth in the end. We want also to protect patients by our accreditation we have in the laboratories and by high quality standards of the test producers. We also want to protect you regarding wrong therapy decisions and damages of your organ system. We need to mention that. I think it's very, very important to say that. Leona, thank you very much. You mentioned already the CAP accreditation, which includes the CLIA, the CLIA, we say. The CLI is simply an allowance. It's not the quality management you do in CAP accredited laboratories. I talked with Bob Mozzaieni with his Bartonellosis test about that, and he's also CAP accredited, and is, wow, we are CAP accredited, we have the highest quality standards, so we are absolutely 1000% reliable, we are 1000% you could trust our results. Yeah, this is the way we need to go in the future. You see the certificates, and Leona, they come every 2 years, the CAP, but they could also come into laboratories and inspect you during that. Without that, you know that they're coming. So they have the rights also to inspect you during these 2 years with them. That's very important to mention that. And this is in the ILAC, as you mentioned, Leona, it's absolutely correct. These accreditation bodies, they are all working together in whole European Union, and in England it's the UKAS, UKAS, which is responsible for your quality. Maybe I forgot one thing to say. This is not just, I'm a laboratory doctor, and I did also a lot of hematology, clinical chemistry, hormones. It's also regarding this, we're not just talking about Lyme disease, it's just about the Lyme disease, which is a really difficult field because of the different test systems. This is all, if you would get a wrong blood count and you would get not the blood transfusion, so you want to rely on that. And this is a quality standard we have in our laboratory as an example. There are some other laboratories fulfilling that criteria. And also the German accreditation bodies, every 2 years we need a re-accreditation. And meanwhile, they come every year to us because this is the modern standard. The next one is in July in our laboratory. And this means really stress. Accreditation, maybe to mention also this point, is not just the analytical process. It's not just the tests we buy from Professor Gilbert or we buy from Euroimmun or maybe other test producers. So laboratories, we always check for the best laboratory tests on the market. It's for all laboratories we are running in the world, I think so, which are accredited. And also regarding this, it's very important to know for you that this accreditation is an international accreditation by the CAP. So I was in Dubai last 2 weeks ago, and believe me, in Dubai, it's not possible to send laboratory tests into not CAP accredited labs. We have also the challenge of the legal aspect. I will come later to that, but you need to send in CAP accredited labs in all over the world, also in Japan or in China. So they all are looking for CAP accredited labs, and because they know exactly for this high standard. And to come back, it's not just what Professor Gilbert is talking about, the reagents or what she's producing according her own accreditation, the test producer accreditation. It's also about the pre-analytics. Pre-analytics, that means which tubes you need for which test and how to take the blood. This belongs all into the complexity of accreditation, temperature controls, etc., logistics checked, how fast it is in a laboratory, and you need to show that. It's not just, now we do some laboratory tests, and that's a huge difference to the LDT, the laboratory developed tests, which are more scientific tests. So the UKAS, it's very important for you in England. This is a certificate, and also you're not allowed to send samples from England into laboratories which don't have the UKAS accreditation or this equivalent accreditation. So please ask your doctor, are you sending into a laboratory which has the UKAS allowance also, because it's illegal to send samples into laboratories which don't have this UKAS allowance or equivalent. You're in England, not in the European Union, so you're outside this ILAC as an example, but they cooperate more and more, and they really check where you send the samples, So because they want a high quality, and also for sure, you authorities in England are not interested in false results or where doctors also and patients could be misleaded and patients could be damaged, and this is pretty clear. But the baseline for this UKAS is in Germany a German accreditation. So if you don't have the German accreditation, you cannot do British samples processing. And I say, okay, please get your money back for that, because simply it's not the quality you expect in this. So please ask all of your doctors, where do you send it into foreign countries? Do they have this European accreditation, and do they have the UKAS allowance or this paper from UKAS? You could get it from us. It's very, very important for us also in this quality management that we, as Leona mentioned already, we have a regular participation in inter-laboratory comparisons for all tests offered. What does that mean? If you develop your own test in your laboratory, there are some tests own developed. I don't want to name them because then you would say, okay, he's against this test. No, but we name them laboratory developed test, LDT. This is more on a scientific level. This is not the quality as Leona Gilbert with her company is producing CE certified test and IVD register test. Okay, this costs also a lot of money, to be honest. The test producers, as Leona is, with her company and others. They invest a lot of money. As an example, we have 4 people working the whole day in quality management. Other laboratories don't do that. They sell the test, they do it without any external control mechanisms. How can you trust these tests? Independently annual assessment of all processes and procedures are controlled. I mentioned already. The obligation, we have the obligation, please, in our laboratory as an example, others also like Mozzainis laboratory, we are just allowed to use IVD registered test methods. For us, it's impossible to do a phages test as an example. We cannot do that because it's not IVD registered, and we would be not allowed to send any samples there, you know. So this means 1,000 quality assurance for you. This is your health, and if you don't get the correct results for your doctors, you get again a wrong treatment, and this will damage you. And so I want really to do a warning about that. It's really also important, as Leona mentioned, validation. Yeah, this test is not validated. This is not correct. A laboratory, as we are running here in Germany and other laboratories with this CAP and European accreditation, we are forced to validate all test results, and the test producers, before what is Professor Gilbert doing in a test producing company, they also, as you have heard, they validate the test clinically, and also the reagents. They need to test and they need to show the authorities. We have the same, so you have a double validation in both if you are using the right and correct laboratories which have the UKAS allowance and based on the accreditation of the European Union. So we are a little restricted in this, and but this is the future of laboratory medicine. You will see we validate them all. We show the authorities when they come to us. Interestingly, the CAP, the CAP, has accredited our Borrelia elis spot as an example, but not the German authority, German accreditation bodies. Why? Because in Germany, they say chronic Lyme doesn't exist. In America, it's a little different. So we have a CAP accreditation for all of these elis spots, which is in Germany the exclusion criteria for Borrelia elis spot. But I think they will find together and will say, okay, it's the same method, it's the same antigen. Why should we give here an accreditation? Why they're not? This is more political discussion we are doing in Germany now. Also very important, as I mentioned, it's not just talking about the analytical process. The pre-analytics is around 60% of the quality. If you, as an example, you have the wrong tubes, or you shake the tubes, or the cells are damaged for the cellular analytics, or it's hemolysis in it. Also, we cannot, as an example, do the hemolytic samples for the tick-plex test from Professor Gilbert. We are not allowed to do that because there's strong regulation. Other laboratories without accreditation, they can do that. You know, nobody's controlling that. But if we would do that, and we mention that also in the test report, you will find hemolysis. Sorry, it's not possible to do that. Hemolysis can happen. We cannot see in the laboratory. So 60% of the quality is done in by the nurses, by the doctors taking the blood process. Also, room temperature plays a role for sure. Refrigeration, frozen temperature. But we are all control checked. You know, we are not allowed to analyze any samples which are damaged. So it's impossible to say, oh, it's a damaged result, wrong positive, because they're not fulfilling the logistics. So this is politically against accredited labs like us. So the freeze store stability is very important for us. We have temperature controls. We track the samples. That's very important. The cell vitality, as you know, these ELISpot tests, cellular tests are very sensitive to temperature. And this, for your safety again, for getting the reliable results, please just send into accredited laboratories as a baseline for your UKAS in England. All other tests, neglect them and say you don't have the accreditation, you're not controlled for that, and don't do that. So the stimulation convalescence of all lymphocytes, we have positive and negative controls, and also very important, we come now to the point, we also compare results with other labs. We are not the only laboratory in Germany doing ELISpots. I think there are 20 others doing. So we send samples, blinded samples from the test producer. It's not Professor Gilbert, it's a different test producer. We get blinded samples, and then we need to analyze what is in the sample. Is it positive or negative? So in laboratories who don't have the accreditation, they don't need to do that. There's not any external control checks needed for that. They can do that, but they're not forced to that. And if we have some deviations, also we need immediate action to preserve the sample integrity for you. The same happened with SARS-CoV-2. If you would get a false positive SARS-CoV-2 swab result, the same situation. Also, we were accredited for the swabs in the laboratory. So this comprehensive control and documentation ensures you, patient, that your samples arrive safely and in optimal condition, meeting the highest international standards. Believe me, you cannot do more than CAP, a double accreditation with allowance for UKAS. It's internationally not more possible. And now, interestingly, also in America, there's a new FDA regulation. Why? It happened in April 2024, so nearly 1 year ago. The FDA, you could be against FDA. You could say they don't want chronic Lyme, but this is independently from that. That means just the Borrelia-Burgdorferi test or other tests for co-infections in our field. There was a final rule to ensure the safety of LDTs, of these laboratory-developed tests, which are not our tests in the accredited laboratories. These are the scientific based. I think, Leona, when you started with developing your, what was it, the tickplex with these round bodies, you also developed an LDT in the beginning, and after that you registered it and you showed the reliability, how to trust it, and then you did this accreditation process, which costs you a lot of money, needed a lot of time, and then you offered your test, and this is exactly the right way. And now FDA forces also all American labs in phase by phase. Now they need to show in May also that is medical device reporting. They and also complaint. If you complain in America, well, my test result is wrong. Also, these LDTs, these more or less scientific tests on a scientific level, offered as routine analysis tests in some laboratories. They say also we need to correct and remove the report, and they need to show the authorities, for the safety of American patients. Then there's next deadline, registration and listing of that, labeling, etc. And you see in the end, in 2028, that's the same baseline also for European tests. Whatever tests you do in the European Union, they all need now an IVD registration, and this is very cost intensive. A lot of tests will vanish from the market. You will see that maybe we in 3-4 years we cannot do any anaplasma test because nobody is doing certification on that. So how can we do that? But in the end, if you go the way of laboratories with this accreditation and with the test producers behind that, you're safe for that, and you could be safe as an example for the ELISpot. And I know Professor Gilbert also is on the registration process, not to lose that you could still do with the tickplex basic or tickplex plus in the future. So this is the way also in America, and it's really high discussed. We could be against this or pro, but in the end, you patient or your therapist, you need to protect your patient, and there's not any discussion that Armin Labs is not reliable or whatever what is said to laboratories. This is just political. And also, if you have lawsuits and you try to win them, if you don't send to an accredited laboratory with CE certified and IVD registered test, there's no chance to win anything. So we supported some lawsuits already. And thank you very much for your attention.
Thank you very much, both of you. Superb information, very, very helpful. We've had several people, doctors, therapists, asking in the chat whether it might be possible to put the slides into a documentation that could be used to actually take to GPs. So maybe that's something that we can discuss afterwards. I'll just read out what one of the doctors wrote to us. It would be wonderful to have a template letter using the information presented by Leona and Armin for patients to use to give to GPs to strengthen their argument to get their tests acknowledged by the NHS, etc. The location of the author won't matter. So that's a
So
lovely idea, isn't it? Because just going in with a pack of slides is going to be a bit harder.
I have it. I have it. So if you just
Oh,
if you contact me, yeah, I can put my email in the chat. If you contact me, I can give you the templates. I've been using it for the Australians and Canadians and now here. So
oh,
I do have a
brilliant.
template.
So could you send that to us, Leon?
Yeah, I
Maybe
can.
we can put that onto the website. That would be fantastic.
Yeah, no problem.
Thank
Let's
you.
see.
And
Yeah.
then another doctor here asked, how accurate are the tests for chronic reactivated Lyme? Diagnosis is notoriously difficult compared to the acute illness.
Mhm.
I don't know
So,
which of you would like to respond to that.
well, reactivation or chronic Lyme. I mean, I would ask, are you trying to differentiate between chronic Lyme and then a fresh, Borrelia infection? Is that, if that's the question? No test is going to be able to do that, but we do have a test for chronic Lyme. Okay, and that's the use of the persistent forms. So by just, following, the titers of these antibodies against the persistent forms and also the spiricate forms should differentiate and help in distinguishing actually chronic form or acute form.
Thank
Yeah.
you. Armin,
So
anything to add to, go ahead, sorry, Leona.
yeah, no, I just saw a comment in the chat. If I can just reiterate one thing here about UKAS. UKAS will accept, because they have signed the ILAC agreement, will accept, okay, any other accrediting bodies certificates from any foreign other lab. That means UKAS has signed ILACs, DAX has signed ILACs, so DAX, which certifies Armin's lab, okay. So UKAS should accept, okay, and demonstrate actually the equivalence of Armin's lab's accreditation. So they're equivalent. And that's the point that UKAS has even provided this letter to Armin's lab that they demonstrate equivalence to UKAS standards. And
Yes.
this is how this ILEX recognition agreement should work. If you're accredited by these accredited bodies that follow and signed on this agreement, then the equivalent standard is demonstrated.
Yes, one could print that out and take it along with the template you've just mentioned. But sorry, just to go back to the chronicity
Yeah,
question,
sorry.
Armin, did you
Yeah.
want to add anything to that?
Yeah, I have, yeah, sure. I worked a lot also with Western blots, immunoblots in my old career. In the beginning, we had the IFAs, interestingly, and they were cross-reactive with syphilis spirochete.
Yeah.
So there was no ELISA. That was the end of 80s. It was Freddie Mercury time. The ELISA came then, and then the Western blot came later on. We didn't do the immunoblots for specification. This is just a story about that. But titer, this is the right word, Leona. We need we have different forms. We have a chronic Lyme patient for sure. We have or name it chronic persistency. Some of the microbiologists discuss about the wording. We have the reactivation and we have the reinfection. So all of these could give us a message in the IgM variation. So you could see in the Western blot as an example more bands, new bands you didn't see before. If you have a previous result, that's always the question. If you don't have a result, you could compare the bands. You could compare the tick-plex results, IgM, what Leona mentioned already. What is independently from that is a ELISpot test. In the ELISpot test, this doesn't matter because it reflects the last 4 to 6 weeks of an infection. So the lymphocytes, they start early in a reactivation to come back in the interferon gamma Th1 immune response we discussed last week. So you have the chance by ELISpot, a unique chance to find out, is it a reactivation? And we discussed last week also about long COVID and reactivation of Lyme disease. And I think, Gillian, it was the IgM, they documented that in this study. So you have options for sure to find out a reinfection, reactivation. Old sentence, Professor Gilbert, one antibody test is no antibody test. I learned in my former time, I could mention SYNLAB, a very huge group I worked years ago. And they said one antibody test, no antibody test. You always need a follow-up of this, an IgM, seroconversion, more bands, more higher titers. Is this correct, Professor Gilbert?
Well, I think it depends on the chronic patient, because I always go back and think about the fitness of the immune system in that patient. Okay, because no matter what happens, okay, ELISAs are going to fail, and ELISA spots are going to fail in these patients if they have a compromised immune system. They're going to fail. So, but in a traditional setting, okay, where the immune system is working, then they should follow, as the immune system should. Okay, we have IgM going up, IG, and then going down. We have IgG going up and then going down, and there will be a basal level, okay, of IgG circulating in our system. Okay, and that's same with T cell. Okay, T cells have memory, so the point is that they should not be at high activity levels, if there isn't an infection. Okay, and that's the point that we have to keep in mind, that when we're dealing with chronic patients, chances are, most likely, and it's been documented, that these people have compromised immune systems. So let's use the test kits wisely on these individuals, okay? And let's look at their symptoms more. Let's look at other biomarkers, because there are other biomarkers to demonstrate chronic patients and so forth. So that's how I would say it as a scientist, okay? That's keep in mind that the immune system is compromised traditionally in these patients, chronic patients. I'm not sure if I answered your question, Armin, but
yeah, thank you very, very much. We've got lots and lots of information on that. Obviously, we'll keep digesting it. Hannah writes here how useful the information was, and thanks you. She does say the issue is the next stage too, as nice treatment isn't sufficient. Even if you get past that stage and they do accept your tests, the treatment that's offered isn't sufficient. So there's further work needed there. She does have the experience that NHS generally don't accept chronic Lyme, but hopefully with these arguments, the templates, the photocopies of the letters, they will. Let me just, we've only got very little time left, so let me just go on to another couple of questions. There was one here. How can patients ensure that their Lyme disease test was conducted in a laboratory that follows ISO 15189 standards?
Ask them. It's a legal requirement that they have to disclose this. They have to disclose their accreditation. And same with the test kit itself. They have to provide the instructions for use, the legal document of the performance of that test kit. It is a legal requirement. Okay, so the instructions for use, just ask them. I've been asking about new tests that are on the market. You have every legal right to get it.
Okay, thank you. And a question that came up in the Q&A earlier. Oh, you've already answered that one, I think, Leona, because that one's gone now. Within the instructions for a range of tests, there are indications that those who received early antibiotic treatment or who are undertaking antibiotic treatment at the time of the blood draw may not mount an immune response. Does this equally apply to tickplex as it does for tests such as the Western blots, etc.?
Well, again, for my test, we've tested this, okay, and it does not matter with the antibiotics treatment on our test. As for the other Western blots, you have to go and look at their instructions for use, okay, and find out that information, okay, if they have actually demonstrated that, okay. There is, I think, 1 or 2 papers published that indicates that delayed antibody response has occurred in patients when right after the antibiotics, but that doesn't apply to all the test kits. Okay, it doesn't. It's what that scientific study demonstrates. It does, like I said, not apply to all test kits. So for tickplex, it doesn't matter. Okay,
thank you. And Armin, anything to say there regarding whether it applies to the non-tickplex tests?
Yeah, this is always the question. Could I use corticosteroids and do the test as immune suppressive remedies, That's a good question always. I want to do an antibody test. I have a strong immune suppression. I have a chemotherapy, or so I have a cancer in parallel. Could I do a test? If a test producer, as it is with the tickplex, would know that it need to be written into the standard procedure protocol, and also that we are not allowed to do the test. Hemolysis, as an example, in the cell is a pre-analytical thing. You're talking now about immune suppression. I would test this patient more for subclass deficiencies, immunology, IgG1, IgG3, if they could produce antibodies or if they could produce T-cell immune response. This is immunology, and this is, I think, more important than to say I test the patient during antibiotics. I say do it, and then we see we have positive controls, we have negative controls, so we double check all of the tests.
Yeah, I agree.
And then maybe one or two more questions just before we finish. There's a great excitement, Leona, that you might have been at the Australian Lyme Centre inquiry. Is that true? Were you there?
Yeah, I was there.
Amazing.
I helped the Lyme Disease Association of Australia and the Lyme Sarcosis organizations with their submissions. I helped other people with their submission. I had my own submission, and then I was invited by the senators to come and give evidence. And I went there personally because I think that was more powerful. And so I also brought this up, these standards and the accreditation process of labs and test kits, and that's why I'm quite prepared for this lecture, is also the template of of what other people have asked, because this is exactly what I provided also to them, because it's true that Lyme disease does not exist in Australia. I mean, that's a wrong narrative. It does exist. And so we're trying to help the patients there and the clinicians as well, because they are scared. Okay. They are being prosecuted against, like we are here in Finland, they're being prosecuted, for treating Lyme disease or even handling chronic patients. So the situation is not just a one country problem. It's really a global problem, and I'm trying to, with colleagues, tackle that with this, changing the narratives and so forth. So.
Oh, thank you. Maybe just one further future-related question, and then we will need to finish. Do you foresee any updates or changes to these ISOs that you've mentioned this evening that could further improve Lyme disease diagnostics?
I'm not sure about the ISOs because they just went through a change. Like I say, the 13485 2016 standard that changed actually quite a lot. But there are other ISOs that may be implemented, like with AI technology, as well as traceabilities of standards, control standards, or risk assessment ISOs. So there's not only these two, but there's other ISOs that TICFlex adheres to, and probably Armin Lab as well. I think maybe they will have some smaller updates, but not very significant ones at the moment. But you never know about FDA in the United States, how that they might influence actually some of the ISOs. Let's see. But even FDA has actually hinted even 10 years ago that they're trying to adhere more towards the 13485 standard, ISO standard for their diagnostics, but they're not there yet. But so maybe acceptance of ISOs, more ISOs, not necessarily the change of the ISOs, I think we'll see in the next 5 years.
Oh, well, amazing. Thank you very, very much. We're considering doing a sort of part 2 of this, where we would like to answer your further questions, because there are quite a lot more. There are some that came in sort of before the webinar as well that we haven't yet answered. So we will be making a copy of the chat and ensuring, and the Q&A as well. Some excellent questions have just come in there as well, which we won't have time to answer. So we will keep those and answer them the next time. Thank you so much, both of you, for your huge expertise and tremendous information.
Yeah, but thank you for the audience's time. Again, it's in the evening, so we really appreciate you coming here and being present. Thank you, guys.
Thank you.
Thank you.
And thank you, everyone, for being there. Just to mention that there is an upcoming webinar, the next one on the 26th of February at 7 p.m. That's another Wednesday. And that will be Dr. Sarah Myhill, a doctor who you all know and love, I'm sure. And she'll be talking about the topic mitochondria, not hypochondria, diagnosing and managing mitochondrial dysfunction and disease. So please do register for that. There is a link actually, which Claire's put up for you to register, but it's also on our events page. at aonm.org. So thank you very, very much again, both of the speakers and for everyone being here, and look forward to receiving your template, Leona,
Yes,
maybe
yes,
adding some of Armin's information to it and
yes.
utilizing that. Fantastic.
Thank you, guys.
Bye-bye.
Thank you.
Thanks very much
Bye-bye.
indeed. Bye.